So good afternoon, everybody, and welcome to today's live webinar. Today we're going to be talking about lessons learned from the 2024 HRD Testing EQA Pilot EQA scheme. Next slide, please. So before we get started, just a number of housekeeping things to address before we get into the detail of the webinar. We've automatically muted your microphone to avoid any background noise. We are recording this webinar, so it will be made available to you via the EMQN and IQN Path YouTube channels, probably 2 weeks or so after the webinar is finished. Most importantly, this is an interactive webinar, so we encourage you to submit your questions via the Q&A feature at the bottom of your screen. There is a dedicated Q&A session at the end of the webinar. Next slide, please. Your feedback is also important to us because that's what really helps make sure that we're providing good quality or high quality training to you as healthcare professionals. So after this webinar is concluded, you will receive an email with a short link to a feedback survey. Takes less than 2 minutes to complete it. Please do contribute your feedback to that survey and to ensure that we continue to provide high quality webinars and training to you in future. So thank you in advance for your support in that process. Next slide, please. So just to go through what the objectives of this webinar are and what you should hopefully gain from joining and supporting it. We're going to give you an overview of the results of the 2024 Pilot EQA scheme for HRD. That's going to include some clear examples of the errors and pitfalls we've identified in that process. We've also got some suggestions for practical solutions which, when implemented, may help prevent or rectify common issues or errors. We're going to give you some guidance on reporting results within different clinical contexts, and most importantly, an opportunity for you to ask questions in the Q&A session. Next slide, please. So the first part of the webinar is an introduction to HRD, and I'm delighted to introduce my colleague, Dr. Nicola Nomano, who's the president of IQN Path and scientific director of the IRST in Italy. Nicola is an eminent oncologist in this field and has done lots of work in HRD. So I'm delighted that he's going to be able to give you the overview of HRD in ovarian cancer. Nicola, over to you.
Thanks, Simon. Thanks for the very nice introduction. These are really very few slides in which I will try to give you some insights on the relevance of HRD testing in ovarian cancer and also the challenges that we have in performing this type of test. May I have next slide, please? These are my disclosure. Next slide, please. So we are discussing today about the relevance of HRD testing in the context of ovarian cancer. Ovarian cancer is the third most frequent cause of cancer death in women with years between 30 and 50 years. This tumor is usually diagnosed in advanced stage, and the relapse occurs in most patients within 2 years after platinum-based chemotherapy. Those patients who are sensitive to platinum are also highly sensitive to PARP inhibitors, and a number of clinical and preclinical studies have clearly demonstrated that the deficiency of the homologous recombination repair pathway is the biomarker of response to PARP inhibitors. May I have the next slide, please? Now, the homologous recombination repair pathway is a quite complex pathway that is involved in the repair of DNA double-strand breaks and interstrand crosslinks. Deficiency of this pathway has been demonstrated in several different tumor types, including breast cancer, ovarian cancer, prostate cancer, and pancreatic cancer. Now, the main cause of HRD deficiency is mutations that can be either somatic or germline of either BRCA1 or BRCA2 gene. So when we find a BRCA1 or BRCA2 mutation, bona fide, we assume that the tumor has an HR deficiency because this correlation has been demonstrated clearly by a number of studies. However, this is a complex pathway, so we might expect that alteration in other proteins involved in this pathway may lead as well to HR deficiency. And indeed, the fraction of ovarian cancer that have HR deficiency, a phenotype of HR deficiency is much larger as compared only with the BRCA mutant. However, there are many tumors that have a clear HR deficient phenotype, but not carry any mutation in any of the HRR gene, which suggests that non-genomic mechanism might lead to HR deficiency as well. On top of this, there are tumors that carry mutation in HRR genes other than BRCA, but do not have a clear HR deficient phenotype. And this most likely is because the role of this protein, some of these proteins is not crucial or somehow can be supplemented by other proteins. But this observation leads to the huge problem how to identify those tumors that are HR deficient and therefore those patients who can really benefit of treatment with PARP inhibitors. Next slide. Now, the biomarker that we use to identify those tumors that have HR deficiency in absence of BRCA mutation is what we define as a genomic scar. And to illustrate, explain what is a genomic scar, I usually show this slide, in particular the panel on the left that compares the genomic alteration in high-grade serous ovarian carcinoma and glioblastoma multiforme. In glioblastoma, the genomic damage in terms of deletion and amplification is localized in specific region of the genome, whereas in high-grade serous ovarian cancer, which has a very high rate of HR deficiency, as you see, the damage, the genomic damage is diffused along all the genome. This is a phenomenon that is referred to genomic scarring, is due to the high genomic instability that's typical of tumors with HR deficiency, and this is really the biomarker that we use for identifying tumors that have an HR deficient phenotype. Next slide, please. Basically, all the tests that we use in our routine clinical practice are based on the same algorithm. We test for tissue BRCA1, BRCA2 mutation, and if any variant in BRCA1 or BRCA2 is identified, then the tumor is immediately classified as HR deficient. In absence of BRCA1, BRCA2, we look for biomarkers that are associated with genomic scarring, less loss of heterozygosity, telomeric allelic imbalance, large-scale state transition, but also other biomarkers. And usually these tests have a kind of score, and if the score is above the threshold, then the tumor is classified as HR deficient. If it's below the threshold, then the tumor will be classified as HR proficient. We do not expect that will benefit at a large extent of PARP inhibitor treatment. Now there are two tests that have been approved by FDA, the Myriad Genetics My Choice and the Foundation Medicine Foundation 1 test, but there are a number of commercial tests and laboratory developed tests that are usually used in many labs to test for HRD. In particular, next slide, please. If we look at those commercial tests based on target NGS, we have many assays that are specific for HRD testing, but also assays for comprehensive genomic profiling that will provide also information for HRD status. Now, in this scenario, it's clear that harmonization, standardization of tests is really important due to the high number of tests using different platforms, different biomarkers, different bioinformatics to assess HRD. Next slide, please. There have been a lot of studies. I'm just showing studies from my lab that have assessed the concordance between some of these commercial tests. For example, in this study, we assessed the concordance of TSO 500, OCA+, and SOPHIA with the Myriad My Choice, which is actually the reference test that we use in most of our studies. And these studies show there's a pretty good correlation between these tests. However, still, there is the issue to have the adequate harmonization and standardization of the process of testing, but also of reporting of these tests. And for this reason, that the International Quality Network for Pathology decided to launch this initiative of an EQA HRD testing. And we asked EMQN and Simon Patton, who is the CEO of EMQN, to lead this project. And we have very interesting results. I'm happy now to introduce Simon Patton, who will share with you the results of this program that I hope will be interesting for everyone, and then we'll open a very important discussion on testing for HRD. Simon, the floor is yours.
Thank you, Nicola. Next slide, please. My disclosures. Next slide, please. Okay, so as Nicola said, so the role of quality assurance is kind of, sort of essential in terms of establishing new clinical diagnostic testing. And the evidence from external quality assessments, not just EMQNs, but many other EQAs, I've quoted 5 different papers here, but there's plenty of other evidence out there, shows that the introduction of any new test is often accompanied by a very high diagnostic error rate. So up to a quarter of all samples tested may have the wrong result for them. And it's undoubtedly true that poor quality testing can or may result in patient harm. And the use of EQA as part of the oversight process can help drive harmonization and improve the quality of the testing process in general. So next slide, please. Next slide, please. Next slide, please. Next slide, please. So if we look at it in the whole as a total quality system, EQA is just one component of a number of different processes that are essential to improving harmonization and driving quality of the entire diagnostic process. That includes the educational component and the use of best practice guidance and training, the internal assessment processes that you as a laboratory would put in place to validate a test, the use of reference materials, and the quality management system that underpins everything that you do. And finally, external oversight of that process, including EQA and the accreditation process as well. And if all those components are in place, then it's very likely that the output of the laboratory will meet a consistently high standard, and therefore you're less likely to generate errors that could cause patient harm. Next slide, please. So as Nicola said, I'm reporting the results of a pilot EQA scheme that's been run by the International Quality Network for Pathology. And Nicola and the team invited members of the IQM path, the EQA members of IQM path, to contribute to this pilot scheme. My company EMQN was responsible for managing the project, but there were 6 partners in total. So CBQA from Canada, IOM from Italy, RCPA from Australia, GenQA, who are based in the UK, and Gentis, who are a French EQA provider. So between the 6 of us, we developed the EQA scheme, and I'm reporting the results on behalf of those 6 organisations on the project that was supported and set up by IQM Path. Not to also forget that obviously this EQA activity was supported from an educational grant from MSD and AstraZeneca, for which we're truly grateful. Next slide, please. So the aim of the pilot EQA was to establish testing accuracy and clinical reporting of HRD testing using commercial assays and in-house developed assays. As Nicola said, many of the assays that started out were purely commercial, but laboratories over the last couple of years have been using and developing their own in-house assays. It's therefore important that we assess how those assays perform compared to the commercial assays. And we are looking to improve the clinical service by using a combination of assessment, so our assessment and feedback to you, the laboratories, via expert commentary. And we do this by a number of different ways. We feedback to each laboratory individually in a bespoke format, tailored to your results in our individual laboratory reports. And we also provide a comprehensive EQA summary report that really summarizes the entire EQA scheme, how everybody's done, all the issues with the different technologies, and the feedback associated with how the labs have interpreted the results from the test process. The format of the EQA was pretty common, pretty consistent to all other EQAs we've run and all the other providers run. So we're really looking at whether the laboratories can determine the correct genotype of the sample, so the analytical process of HRD testing. We then look at the interpretation of those results in response to the clinical referral, so making sure that you're able to feed back the right interpretation of the analytical result. We're also assessing whether your laboratories are using the correct internationally accepted variant nomenclature. We need to have a standardized way of reporting the variants that we may or may not find within the samples that have been tested. And also, fourthly, we're looking at how we provide accurate patient information and sample identifiers. And also actually how the report is laid out and conveys the information back to the referring clinician. Next slide, please. In common with many of the pilot EQA schemes that have been run, the first part of it, all these processes, we're developing it for a completely new biomarker, is really understanding the state of the art. And so we ran an expression of interest survey and sent it out to all the laboratories within the network of the 6 EQA providers. We had 73 responses, and as you can see, nearly 70% of those responses were from laboratories based within Europe, but we had feedback also from Asia, Africa, North America, South America, and Oceania. So looking at the data from the 84 laboratories, 28 represented 28 different countries, and you can see the representation of the numbers of labs in the figure on the right-hand side. Next slide, please. From that, we then selected the laboratories that we wanted to participate. So this is a pilot. What we're really trying to do is test the EQA process. So we actively selected laboratories that were running a clinical HRD service, and that there was a broad representation of all the different testing technologies, both in-house developed tests and commercially available assays within the EQA. This really allows us to get a feel for whether the format of what we're trying to do with the EQA process is going to meet the needs of the laboratories and allow us to really dig down into the detail of how the analytical process is working and how the reports are actually being authored and sent out to the clinicians. So we broke down the number of laboratories by the different EQA organizations, and proportion them based on the number of laboratories that had completed the survey. And you can see that in the end, we had about 50 laboratories in total across all the 6 different EQA providers. From that, there were a number of laboratories that didn't submit results. So 6 laboratories in total. Which is a shame, but not unusual. Obviously, those 6 laboratories then deprive other laboratories that may have wanted to participate but couldn't, because we decided to accept certain laboratories. So that's a shame. But I think at the end of the day, we had a reasonably consistent number of laboratories that allowed us to really test how the EQA performed in the real clinical setting. So on the right, you can see the breakdown of laboratories that participated in the EQA scheme. And you can see that it was a pretty broad representation across all the different EQA providers' territories. Next slide, please. The samples we used for the EQA were 3 commercially manufactured FFP samples available from LGC Seracare. They had already been validated using next generation sequencing by LGC Seracare, but consistent with all the EQA schemes that we run, we also independently validate them before we send them out to the laboratories to be sure that we're happy with the way they are performing, but also that we've tested them in a range of other different analytical processes. So we use 2 different validation laboratories using different testing methodologies. Both of them were commercially available tests. And that allowed us to develop to gain a little bit more insight into how those samples performed in a number of different tests, not just the ones provided by the manufacturer. The samples themselves, the 3 different samples, we had one sample with a genomic instability score of 72 plus or minus 3, so a level of uncertainty there, which was being classified as high positive, so HRD deficient. Then once that also contained 4 variants in the ATM gene, BRIT1, RAD51C, and RAD51D. Then a sort of intermediate level sample, if you like, that was around about the threshold for genomic instability score, so low positive, which also contained a BRCA1 mutation. And finally, a sample with a low genomic instability score, which also contained variants in the 4 genes shown in the first sample, but this time at lower variant allele frequencies. Next slide, please. Looking at the assays used by the laboratories in the testing process, so breaking down by approach, there was 1 laboratory using a microarray approach. 36 laboratories, or 82% of laboratories, used a commercial NGS-based assay. and 16% of laboratories used an LDT, laboratory developed test, based on NGS. And breaking down the actual assays themselves, you can see that the Sophia DDM assay was the largest contributor to that, but there was a broad range of 8 different assays that were used, including LDTs. Next slide, please. From an EQA perspective, in terms of requirements and timelines, we asked all laboratories to perform BRCA1 and BRCA2 testing and to establish HRD status on the samples. We encouraged the laboratories to also report other HRR genes where tested. The data that was submitted via an online results submission form, but we also collected copies of the clinical reports so that we could assess the interpretation and the report layout and format from each of the different laboratories. Laboratories were given 10 weeks to do the testing and to submit the results to the EQA providers. And the assessment process covered the analytical component of the test process, the genotyping, the interpretation, and also the report structure, content and format. The marking and assessment of the results from each laboratory was done against a predefined set of marking criteria, guided by external experts to ensure that we were consistent between all the different laboratories and different EQA schemes. And because this is a pilot EQA scheme, if laboratories made a mistake, there was no poor performance awarded as a consequence, as it's a pilot EQA scheme. Next slide, please. So looking at the samples in more detail, so although I said there were 3 different samples, we didn't obviously include them in the same order. So looking at the details in more detail in terms of the gene variants, you can see for sample 1, this was effectively the HRD proficient sample, and the state, so negative status, and you can see the different variants that were present within the sample, all at relatively low variant allele frequencies, and the fact that there was no BRCA1 or BRCA2 variants were supposed to be detected in that sample. Case 2 was the high sample, so the HRD positive sample. Again, you can see the same genes and same variants, but this time at much higher level variant allele frequencies. And again, no BRCA1 and BRCA2 sample pathogenic variants were expected to be detected. And finally, in case 3, which was the low HRD, so still HRD positive, we expected laboratories to identify a BRCA1 variant in the sample, but no other variants in the other HRR genes. Next slide, please. So looking at detail in the results, so 44 laboratories submitted results, of which 33 also provided a clinical report and therefore an interpretation of the clinical result. From a genotyping level, for case 1, 38 laboratories reported the correct HRD status, and there were 3 incorrect genomic instability score statuses, and 3 samples failed the test process. 36 out of the 38 laboratories provided clinical interpretation. Case 2, there were no clinical errors, no genotyping errors, and 42 labs reported the correct HRD status. 2 of those samples failed in the test process, and 37 out of the 42 laboratories provided a detailed clinical interpretation of the result. Finally, for case 3, so the HRD positive sample, but this time a low sample, 40 laboratories got the right HRD status. 1 laboratory reported conflicting results for the genomic instability score and the HRD status, and so was classified as the wrong genomic instability status. And 3 tests also failed. 36 out of the 40 laboratories provided a clinical report for interpretation. Next slide, please. Looking at the critical genotyping errors, so the analytical errors. So these are errors that we would classify as potentially leading to patient harm. So in a bit more detail, for case 1, obviously there were 3 genotyping errors. In one case, the laboratory reported deletions in BRCA1 and BRCA2. They were using a Thermo Fisher Oncomine test. One laboratory reported the GIS status as positive using an LDT, and the third laboratory reported the GIS status positive, but this time using the Thermo Fisher Oncomine test. The other error that was made was in case 3, where the laboratory did not report the BRCA2 variant, again using the Thermo Fisher Oncomine test. Next slide, please. So looking at the overall performance of the laboratories in the EQA scheme. So the methodology for reporting results back to laboratories is if a laboratory gets all the results right for each category, whether that be genotyping, interpretation, or clerical accuracy, they get 2 marks. And every mistake that they make they lose a deduction from the total of 2 marks. If they get something completely wrong, so in fact they make a critical genotype error, then they get a score of 0. So looking across the 3 cases, you can see that genotyping scores were consistently high, and the clerical accuracy was also pretty high. And unsurprisingly, the interpretation scores fluctuate a little bit in between the 2 of them, because interpretation is far more subjective than whether you get the correct analytical result or not. So 90% of the clinical reports, contain the correct interpretation of the clinical scenario in the context of the prescription of PARP inhibitor therapies. We awarded 4 critical genotyping errors, which is an error rate of about 9% of the total number of samples tested. And there were no poor performances in this as a pilot EQA scheme, so there's no consequences for making that mistake. Next slide, please. So a lot of learning came out of it, and obviously we fed back to each laboratory in detail individually about the learnings we identified from it. But if you look at the summary report, which is available to all the laboratories that participated, we've really sort of summarized them in the next 4 slides, the key learnings from it. So looking at the analytical process, when reporting a continuous score, and obviously the GIS status is done on a scoring system, it's recommended to state that the validated threshold for clinical actionability. And of course, this will differ depending on the different assays, and many laboratories fail to include the predetermined assay-specific cutoff values for the GIS score. Cases 1 and 2 contain several non-BRCA HRR-associated gene variants, and many HRD assays include comprehensive HRR gene panels within the scope of their test. Many laboratories did not report these HRR variants, despite stating on their reports that they were tested. So this is really a question of ensuring that your clinical report actually includes the context of the scope of what you've tested. So if you are testing HRR gene variants, you should really be reporting them on the clinical report. Very encouraging to see, but the use of a standardized variant nomenclature was consistently high across all of the reports from the different laboratories. Something that the EQA providers have obviously championed for a very, very long time. But you can see it in the context of ensuring that we consistently feed back the right results to the audience, the clinicians, that we need to have consistency in how we do that, and the use of a standardized variant nomenclature is essential to make sure that happens. And encouragingly, the EQA materials perform very well with only 7 test failures. So that's about 5.3% of all the tests undertaken in the EQA scheme. This is always a challenging perspective when you're providing an EQA in a very new biomarker, and the LGC samples performed well in this EQA. Next slide, please. Looking at the learnings from the interpretation, so 90% of the clinical reports linked the genotyping result to PARP inhibitor therapy. Remember, the question was asking whether these patients really would benefit from a PARP inhibitor. And it's an important aspect of the clinical interpretation, and if it should be reported if local policy allows, and therefore preventing the opportunities for patients preventing the likelihood of patients from not receiving the drugs that they would benefit from in the first place. So it's great that only 10% of the laboratories didn't include this component of the report, the essential interpretation. Multiple different assays were used, as we showed earlier on, and each of them has a different system for scoring GI. However, it's really apparent that there's still a need for standardizing reporting the results, and it was very clear from all the reports there was massive variation in how people report the thresholds associated with the GIS status. Laboratories use a range of different terminologies, including HR proficient and HR deficient, HRD positive and HRD negatives, and in some instances, GI score was described as HRD score. Even myself, within the context of this webinar, I've used all four of those. So clearly there's a need for more standardization on terminology associated with HRD. And if likely pathogenic and or pathogenic BRCA1 or BRCA2 variants or HRR gene variants are detected, because there is always a potential for a hereditary component to this type of result, the recommendations for confirmatory germline testing and counselling should be provided in the report if the patient has been consented and the appropriate referral has been given. So always remember, if you like, the hereditary component of these types of tests, and ensure that the appropriate follow-up and testing has been carried out after detection of a variant in one of these clinically relevant genes. Next slide, please. So some general learnings from the EQA scheme. So the pilot EQA scheme generated a lot of useful data on HRD, the HRD testing landscape. So we looked at the state of the art before the EQA and the performance of those assays and methods in the EQA scheme itself. Most EQA schemes focus on specific gene alterations that are simple to test and interpret. Obviously, HRD is a very complex biomarker, and the EQA itself needs to be able to meet the needs and challenges for this type of assessment. But it was clearly quite a challenging process for us to be able to assess the performance of this complex test in the real world setting. The assessment process raised a number of different questions on how we might delineate our own marking criteria between genotyping and interpretation categories. In other words, how we actually score the laboratory's performance based on the outcomes of that testing process. And that's something that will be reviewed and improved as the next round of EQA takes place. Further discussions needed on improving and more improved and inclusive marking criteria really feeds into the point above. And as yet, there's no consensus on whether other HRR genes should be assessed if a laboratory tests and reports them. As Nicola quite rightly said, a deficiency or a variant in one of the other HRR genes outside of BRCA1 and BRCA2 does not necessarily lead to HRD. Next slide, please. Some observations. So our pilot EQA scheme was oversubscribed. There's clearly a large demand for EQA for HRD, which reflects a large number of laboratories doing HRD testing. It only gave us, if you like, a snapshot of the HRD testing practices and reporting formats, but it's given us sufficient background and information and to allow us to develop the ongoing EQA schemes to really get a good feel for how HRD testing is taking place in the clinical setting. Overall, I think this is really pleasing to see, given the complexities of the test. There was a very high level of interlaboratory reproducibility. In other words, the implementation of this test in the clinical setting is being done well. And many laboratories don't indicate a GI score cutoff value on the reports. So a validated threshold for a clinical actionability is recommended, and we covered that in the interpretation part of it. The reporting language around HRD status is also varied, and sometimes the GI score was described as an HRD score. Again, not a surprise. This is, as I said previously, part of the issues associated with a brand new test, and the harmonization and driving towards consistency of the test results and how it's reported is part of what EQA is here for. Next slide, please. So we are undertaking a number of different dissemination activities around this pilot EQA scheme. So we've obviously published the results of the EQA scheme for all the participants. That took place in May. And we're also currently writing up the findings in a peer-reviewed journal that's in preparation. This webinar is to disseminate the results of the EQA, but also we've submitted an abstract, and a poster is available, which is going to be presented at the European Congress of Pathology meeting this weekend in Vienna, in Austria. So happy to share that, copy that poster if you are interested in seeing the poster itself. Next slide, please. Finally, just the acknowledgements. So not just to the sponsors of MSD and AstraZeneca, but also to all the EQA providers who put a lot of work into this. So to my team, Arthur and Vic, to the IQM Path team, Nicola and Francesca, to the IM team, Nicola, to RCPA, Tony, Jen and Tis, Etienne, CBQA, Tracy, and finally to the GenQA team, Jenny Fairley and Sandy Deans. All of them contributed a lot of time and effort into ensuring that this pilot EQA scheme ran to time, and has been a comprehensive assessment of how the HRD test process has been implemented in the clinical setting. Next slide, please. Okie dokie. So that's the end of the feedback on the EQA webinar. And I think we are now throwing the floor open to you guys in a question and answer session. So if you do have any questions questions you wish to ask myself and Nicola, please do use the Q&A to put them to us. Nicola, I've got one for you as a starting point. Is that okay?
Yeah, let's go. Let's go. I took a lot of notes, so I have many questions for you, Simon. So while they are coming, I mean, there are many interesting points that I think we should really address in this. And let's start. Okay, let's start. You can start with your question. Come on.
So just looking at the diverse landscape of HRD testing, where do you see the test process going in terms of the use of commercial and LDT tests? Is there any evidence from your work on comparing them directly that one performs better to the other?
Well, I think that at the end, most labs will use a commercially available test. Still, the issue is the validation of this test in terms of clinical validation, Simon, because I mean, the clinical validation should be performed on samples for which we have information on the outcome, and this is what some companies are trying to do. I can tell you that for some tests, there are data beyond, on validation, and other data are trying to be accumulated. But there are some tests that are widely used. I'm not going into details, for which the validation was done maybe on 20 or 30 samples, and without having demonstrating any clear correlation with the outcome. So the issue is that even with the reference test, there are some patients that might still benefit of PARP inhibitors, so we might not be able to pick them up. So it's clear that even with the reference test, the sensitivity and specificity is not 100%, but still we need to compare with those tests. On the other hand, if you don't have a test that are reliable and being validated in a appropriate number of clinical samples, then you really take the risk to give false results. I have to say that I'm pretty happy about the results of this pilot, because this is a complex biomarkers, it's a new biomarkers, and the error rate was quite low. I mean, lower than what I expected. So I think these are encouraging results. And actually, maybe one point that Simon, we missed is that we should also test the reference material with amplicon-based assays, such as the Thermo Fisher, because the validation was done only with hybrid capture. So I think that we should go back and also assess with the amplicon-based, just to understand if there are any limitation with the different technology. But I mean, excellent work up to now. The other point that I think is very important is this story that you highlighted of the scoring and threshold, because each of these assays has a different scoring system and a different cutoff. And I mean, even for oncology, it's very, very difficult. If you read a report to have the interpretation, the cutoff is 42 per million, 16 for Foundation, and I think also for Thermo Fisher is 0 for Sophia Genetics. So I mean, if there is no clinical interpretation and no correct reporting of the results, I mean, the risk is really that the patients might not receive a very effective therapy. What is your thought, Simon?
I absolutely agree with you, Nicola. I mean, the EQA showed that, didn't it, in terms of the results, the inconsistencies between how laboratories report the cut scores and the cutoffs, and the huge variability. I mean, we didn't present the detailed data here, but in the summary scheme report, it's very clear that needs harmonization. And like all these processes, part of it is about how you communicate that back to the end user. So it's perfectly okay to use a different type of test and with different thresholds, but you really need to make sure that how you feedback that to the end user is clear in the report. So you can use differences and have differences in the thresholds, but the report needs to very clearly communicate the scope of the test and what was included and how you came to that result.
Now let's try to give some advice for labs who are starting for HRD testing. The first point I already touched on is that you better, I mean, if you want to develop your LDT, I mean, you are free to do it. If you choose to use a commercial available test, I mean, look in the literature and try to use a test for which there is a background. But then, what is your suggestion, Simon, to set up the workflow and validate, a new test?
Well, I
So
think
complex as HRD.
the validation side of it is a really difficult question to answer. So we've got a question that's come in the Q&A around the use of real clinical samples in the EQA process and why we didn't use them, because obviously we're using artificial reference materials, and that clearly doesn't there are samples that are, if you like, too perfect, and therefore you can't replicate some of the problems associated with sample quality. So sample validation is really essential. And as an EQA providers, we have to use a sample that we can get reproducible amounts of tissue from and be able to send the same sample to everybody. So we started out in this pilot EQA with very, very high quality samples, but we have also done a lot of background work with IQM Path on validating a range of different other reference materials, none of which are real clinical samples, because obviously clearly you can't get enough material to be able to send to everybody, but samples that have a range of different variants and that are present at low or high variant allele frequencies.
Hi, Simon. Yeah, there is a question on the use of the material, synthetic material rather than FFP material from patients. Actually, do you want to address this question? I mean, we should explain why we can't use, in most cases, tumor material.
Yeah, I mean, the clear answer is if you want to send a real clinical sample around to 85 different laboratories, you can't do that with a real sample. There's just quite clearly not sufficient tumor to be able to do that. And they also have the evidence, the issue of tumor heterogeneity associated with that as well. So the only way to provide an EQA scheme that allows you to assess analytical performance objectively amongst every laboratory taking part is to use a reference material, in the acknowledgement that sample doesn't totally represent a real clinical sample. But that's a limitation we have to accept as part of the quality assurance process.
Yeah, it's always like this, the question. But the problem is that at least for these schemes organized by IQM Path, we have 100 of labs participating at the end. And it's almost impossible to send the same tumor tissue to every lab. That's why we opt out for this. And this material is really a bank of reference standards that can be used also by labs for validating their results. I mean, I think that this is what we are trying to build up, not only for HRD, but also for other biomarkers. We have also a question on the high failure rate for Thermo Fisher. We should clear out that the numbers are very low, so it's very difficult to claim there's a high failure rate for the Thermo Fisher. And I think that there was a case in which the BRCA variant was not reported, but the score was correct. So the case was classified as HRD anyway. And I think that we and other labs have described that most of the inconsistent results with thermofish, but also with other assay, occur when there is the value is close to the threshold, which is related to the variability of this assay and to the complexity of the biomarker. Do you have same similar observation, Simon?
Absolutely, Nicola. I mean, I think this is a small subset of laboratories run in one pilot EQA scheme. There's simply not enough data there to draw any strong conclusions on assay performance. And you know, four samples failed, of which 3 were using the Oncomine, but that doesn't mean that the Oncomine test itself has a problem. So I think we have to be very careful about us trying to interpret the assay performance in such a limited data set. We need to run these EQA schemes through 2, 3, 4 times to really get a feel for how the various different assays are performing in the real clinical setting.
And with a much larger number of symbols. And as I told, you know, we didn't validate with the thermofish because at that time, you know, the thermofish was not really a player. Now it's starting to be more and more used also for HRD testing. So we will get back to the symbols and we'll see if in our hands the thermofish is able to detect that variant and the GI score. Any results about the 2025 HRD scheme? Simon, we cannot anticipate anything, I guess.
Not yet, because the samples haven't gone out, Nicola, but the samples go out the end of this month. So the results will be reported.
This is the way in which I was trying to push Simon to rush up, but yeah, it's okay.
Yeah. I thought you might be trying to push me to hurry up, but no, the samples go out at the end of this month. So the results in that EQA will be reported in February, I think. So yeah, still a while to go before we have that data.
Let's address also this issue of the non-BRCA HRR gene variants. What is your thought on this? Should they report it and described? I mean, what's your idea? Then I give you my idea.
We might have differing opinions on it, Nicola, but my perspective is that if you're undertaking a clinical test, you should report all the biomarkers tested within the scope of that test, and that's the approach we take for all of our EQAs. And that's so, you know, state if you did test several HRR genes, you should state which genes have been tested, and if you detected variants in them that are not clinically relevant to the context of HRD, you should still report them, but maybe put them in a section that makes that very clear. So I think you should report on all the genes that you've tested in that test. Nicola, what do you think?
Yeah, I also think, you know, that we know that there is no direct correlation between HRR genes other than BRCA and HRD status, but it's also true that we have a little knowledge on these variants. And maybe for the same genes, for some genes may be more relevant, for some genes, some variants might be relevant. So I think that testing these variants and reporting the variants will be very important in the future to get more information. And then there is also the issue of the possible involvement of some traits of this gene in hereditary cancer syndromes, which is another important result and information we can get from this assay. So I agree that if they are tested, they should be reported and described, although they are not actionable. But I mean, not have, there are variants that are relevant, although they are not actionable. So you should start from this point. I think it's a good idea to report and describe if you are testing these variants. Although sometimes the classification is more difficult because there's little knowledge. We must acknowledge this.
Agreed, Nicola.
Now,
I've got one here for you, Nicola. So from your clinical perspective here, really, is there a recommendation for this from the assessors as to the best terminology to use, i.e. HRD positive versus HR deficient? We could see from the results there was massive variability. What's your perspective on that?
Yeah, it's very, very difficult because the problem is that the end user is the oncologist. And I can tell you that with my colleagues, it's very difficult, you know. So I see that in most cases, they understand more HRD positive. So I would suggest, as we did for KRAS, that, you want to use the correct definition, but maybe you want also to add a clear definition for the, so HR deficient, comma, in parentheses, HRD positive will solve the issue, but actually should be HR deficient, HR proficient is technically correct. Otherwise, HRD positive, HRD negative doesn't mean very much. But I think that most of my colleagues will understand better HRD positive and HRD negative. That's why it's important to give also the cutoff, because if they look at the interpretation, because if they see, okay, it's 15, it's negative, but maybe it's Sophia genetics, so it's with a cutoff of 0. So you want really to be clear that what you are testing, which is the threshold. Now, could you share which commercial tests have been used and which of them are the most frequent? I mean, you showed the data of the survey, but although there's a limited number of labs that participate in the survey, what do you think?
Yeah, so I showed it in the presentation itself, the breakdown of the different commercial tests being used in the pilot EQA scheme. I'm happy, obviously, to share a copy of the report, Nicola, should the participants wish to see a copy of it. And we also have a poster that's been presented this weekend at the ECP, which summarizes quite nicely the results of the pilot EQA as well. Again, happy to share that to all of those of you who've dialed in. Just leave a feedback at the end and we will get back to you.
Okay, now there's a question on the classification. If you don't find any BRCA variant and the genomic stability score, how is it calculated, is low, even in presence of other HRR genes, the case is HR proficient or HRD negative, is what. So it's clear that, I mean, this is the classification. That's has to be clear to everyone, because there's no other way to go, even with IVAF. But if you find a high VAF in an HRD associated gene or in a HRR gene, then the patient might be referred to genetic counseling. You know that there is a list of about 40 genes that for which German follow-up is recommended by the European Society of Medical Oncology. As if you look at that paper, you have an idea of which genes you should really act for genetic follow-up, German follow-up. Is there a recommended minimum tumor cellularity for HRD analysis? This depends on the test, because some tests require at least 30% of tumor fraction, others require, might work even with the lower tumor fraction, but it's clear that lower is the tumor fraction, higher is the probability to have some false negative, in particular for those tests that are, let's say, with the scores that are close to the cutoff. So this is an important issue. So it's very important that there is a preliminary assessment by the pathologists of the sample, if possible, to make a macro dissection, so to enrich as much as possible the samples of tumor cells, because this will improve the quality of the test.
But Nicola, that point is very true, but it's also relevant to the context of any test on an FFP material. The low tumor cellularity is a really key important point, but you should treat these samples like you would do any other test. I know it's a complex biomarker, and therefore there is if you like, a requirement to have a bit more tumor in there. But the point is that this you should treat these samples the same as you would do any other pathology-based samples.
That's true, but when you are looking for a single nucleotide variant, for example, an EGFR mutation, then you push with your coverage and you can find it. But when you have so complex biomarkers, the tumor cellularity will play a major role in having or not reliable results.
But it also comes back to how well you validate your assay and whether you're using a commercial assay or not. So in a commercial setting, it'll come with the limited detection and other associated metrics associated with the type of sample you're testing. So you're right, but it's also a general point of all FFPE testing is that you need to make sure you've got enough material to start with, and that material does contain tumor itself.
Sure. Now, any other issue? I don't know. Oh, the samples were sent out already for 2025. I don't think for American Impact.
That's quite likely they have already gone out. I'm slightly disassociated with the day-to-day operational perspectives. They may well have gone out, but they would only gone out just before the summer break. So yeah.
Yeah, and then there's a question on glioblastoma. Of course, there are clinical trials which PARP inhibitors are used not only in tumors that have a clear HR deficient genotype, but if you test thousands of patients, you will find that there is a patients without breast cancer or ovarian cancer and so on that still have an HR deficient phenotype, and there are efforts to develop this biomarker as a tumor agnostic biomarker, but we are far from this, so this will take time. Any other suggestion on how to improve testing and reporting, Simon, from your side?
No, I think at the moment it's too early day. It's too early. So we've only run one pilot EQA, and we've got some good background data on how labs are doing the testing and reporting the testing. I think we need to see at least one more round's worth of data to really start to draw some stronger conclusions about areas for improvement. What I would say is there is a strong need, I think, for best practice guidelines to help support laboratory testing on HRD. And I think, we're not there yet, but one more round of EQA, we should have enough evidence to start to maybe push IQMPath for some support to do best practice guidelines around it, or to work with other professional organizations to develop best practice around HRD testing. So that, for me, that's really where I think we can start to influence the quality of testing more generally moving forward.
Yeah, actually, the results of these EQA schemes will be very important to understand where are the points in which we need to put more attention, and also where are the areas of improvement for labs in testing, reporting, and so will be very insightful also to prepare guidelines and recommendations that could support and help somehow the labs that want to implement the testing. In this respect, let me say that this international collaboration is really important, and also I suggest to all labs that are participating to, participate to the surveys that come from IQMPath, from its members, because participating to the survey is a way in which you can then participate also to this pilot EQ scheme that can help also, to improve your testing, because indeed this is the way in which we select the labs. We have a new question, I think, a comment. I agree that tumor fraction solarity is very important. Over time, we should encourage molecular assays to report the tumor fraction to compare with the histopathology. That's a good point, but unfortunately, at least most of the assays that are available today do not estimate the tumor fraction, but this is something that should be implemented and be very important for the correct interpretation of the results, and to understand, distinguish from true positive from possible false negative. Okay.
Nicola, I was going to add that, so to come back to the previous point to that, so obviously it's too late to participate in the 2025 EQA scheme. The samples have already gone out.
Okay,
but the registration process for the 2026 schemes opens on the 15th of September via EMQN. So if you want to participate in the 2026 scheme, then keep an eye out for the registration process opening on the 15th of September.
Yeah, very good, because this is it's clear that there are more and more labs, and in particular when you are starting in somehow implementing the test, it's important that you participate every year to an EQA. Then after a while, every other year could be also a strategy, but it's very important that we really keep somehow updated on the technology. If you change the technology or update the technology, it's very important that you participate again to an EQA scheme. Now, Simon, I think that if there are no more, I don't see any other questions, we have no other points. Do you want to say a few words to close this session?
I do indeed, Nicola. I always like to say a few thank yous at the end of it. So yes, primarily a big thank you to you, the participants, for dialing in. Please do try and contribute less than 2 minutes of your time to the survey. You'll get the email very shortly. A big thank you to MSD and AZ for supporting this webinar and the EQA. And big thank you to the labs that took part in the EQA. Obviously, the EQA only works with your participation. And finally, Nicola, a big thank you to you for your time today in the webinar. And we look forward to catching up with the labs maybe next year on another feedback webinar. And as I said, don't forget the EQA opens for registrations on the 15th of September this year. Thank you.
Thank you to all, and have a good day.